Since the discovery of the cell as a fundamental unit of living organism, it was clear that cells need to communicate to survive. Intercellular communication is also fundamental for not only for cell survival, but also in diseases. Cells can communicate by different mechanisms. And recently, tunneling nanotubes, which you can see here in this image, have been emerged as another mechanism of intercellular communication. These are long, thin protrusion based on actin, which you can see here in violet, which allows cells to communicate directly between each other. In my lab, we will first discover that tunneling nanotubes allow the exchange between cells of amyloid proteins involved in neurodegenerative diseases. In this review, we compare the communications between cells mediated by tunneling nanotubes in the frame of neurodegenerative diseases with other mechanisms of intercellular communication, including extracellular vesicles.
So this is still uncertain whether TNTs originate from the differentiation of polypodia or if they're entirely different structures from the beginning. What has been shown is that there's many common actors and regulators involved in their formation, which we discuss in this review. Now this isn't really surprising to us, given the fact that they're very similar structures. At the same time, these structures will serve very different functions. If you think now about extracellular vesicles, these are structurally very different, but they will serve very similar functions. Both TNTs and extracellular vesicles, for example, have been shown to be upregulated in conditions such as hypoxia. And they're both involved in similar physiological and pathophysiological processes, given the fact that they're specialized in intercellular communication and have the ability to transfer material. Now, some actors have been identified to be shared between extracellular vesicles and TNTs. For example, both are positive for some tetraspanins, which are membrane proteins known to be, in some conditions, involved in fusion processes. And also, in the case of ectosomes and TNTs, both have been shown to be formed using the IBAR protein RSP53, which can induce negative membrane curvature. So it's interesting to us to understand whether the pathways leading to the formation of extracellular vesicles and TNTs could be intertwining.
Recently, TNTs have emerged at the forefront of intercellular communication between the connected cells. Different types of cells and of different origins have been shown to be connected via TNTs, which facilitate the movement of intracellular ions, RNAs, proteins, invading pathogens such as bacteria and viruses, of special importance being the recent SARS-CoV-2, intracellular organelles such as mitochondria and lysosomes, as well as aggregate-prone proteins that cause neurodegenerative diseases, have been shown to be spread via TNTs. In the context of the central nervous system, it's not only the neurons that are connected via TNTs, but also glial cells. Such connections facilitate the transfer of these aggregate proteins between the connected cells, as well as could potentially act as a rescue mechanism via which different helpful molecules can be transported to unhealthy cells. As such, TNTs in the field can be considered to be a double-edged sword, which can provide a protective function as well as aid in the spread of pathology. In our review, we discussed the different kinds of aggregate-prone proteins that spread via TNTs, both in vitro and the recent results that have been shown for in vivo conditions.